Hi, I'm Mitchell, and today I'm going to be talking about why biology is the most significant science in terms of advancement in the past 100 years. So, one of the first examples I have is the development of penicillin and other antibiotics. Alexander Fleming was the first person to discover an antibiotic, which was later termed penicillin. He found it when he was using petri dishes in his lab, and he realized that there was a mold juice that killed a wide range of bacteria. And after more testing of this, because he thought it was such an interesting phenomenon, he realized that this mold juice could kill almost any bacteria, especially bacteria that cause disease. And after this was introduced, after this was developed, it started to be used and tested on diseases, which eventually led it to be mass developed in 1940s due to World War II and a lot of infections that people would get from bullets. And it is estimated that antibiotics have saved up to 200 million lives because they're effective on almost any bacterial infection, which has significantly reduced the amount of people who are dying from these types of infections. And even though antibiotics have run into challenges such as mutations from different bacteria to resist them, more and more antibiotics are being developed to the point where almost any bacterial disease can be treated with antibiotics. And another major development in biology was the discovering of restriction enzymes. And restriction enzymes were discovered by Werner Aber, Daniel Nathans, and Hamilton O. Smith. And these are enzymes that can cleave DNA molecules at or near a specific sequence of bases. So basically, what they do is they kind of help with the replication of the DNA code and to continue the development of bacteria. And what they realized is that these restriction enzymes could tell different cells what to produce and what to do because they kind of run the DNA and the replication of it. So what scientists began to do was they would tell different bacterium, such as E. coli, to produce things that they normally wouldn't produce, such as insulin, which is usually done by the pancreatic cells. And this is a massive discovery because diabetes is one of the most significant diseases on the planet, especially in first world countries where these type of treatments are affordable. And what has happened is they create insulin from E. coli, which allows for these people with diabetes to live longer. And another big, more recent biological invention or discovery is that of cloning. And one of the most significant examples we see of this is with Dolly the sheep. Dolly the sheep was first cloned on July 5th in 1996. It was caused when it happened when a female sheep cells were placed in an unfertilized egg, which allowed Dolly the sheep to be born. And it proved that cloning was a possibility, which many thought was more like science fiction, like it couldn't happen realistically in the future, even with thorough testing. So what it has done is it has allowed us to advance with science, knowing that cloning is an option if we ever would necessarily need it. And it's not 100% proven yet, but it's a major stepping stone in the terms of reproduction in the future. And another extremely significant biological discovery is that of defective genes. So when mapping the gene code, especially during more recent time periods, scientists have begun to discover that certain genes allow for certain diseases that are more genetic. And one of the first one to be discovered was that of cystic fibrosis, which was found on chromosome 7 in 1989. And this first discovery allowed scientists to discover that there was a lot of diseases that were caused by genetics, and through the mapping of the genome, they have been able to find certain chromosomes that link to diseases such as sickle cell anemia, Huntington's disease, and Down syndrome. And it also has allowed babies to begin getting scanned for these defective genes. And it can also allow parents to get scanned to see if they're highly likely to have a baby with one of these defects. And this is revolutionary in terms of helping people discover whether or not they'll have these illnesses early. And that way they can get in programs that'll help them deal with these diseases, especially mental disorders, because if you are able to learn and teach these different children earlier, they can be more effective in their future. And it also helps the fight for a cure because we know that in order to cure these diseases, we have to fix the genome sequences rather than medication, because medication won't fix the genes that are causing the issues. And that's the end of my presentation. Thank you.